Welcome to this Fast Facts presentation from BioInsights and SK Pharmteco. I'm Lauren Coyle, a commissioning editor at BioInsights, and today we'll be looking at accelerating time to clinic for advanced therapies with a platform approach. Joining me today are our speakers John Lee, Cedric Rousseau and Dana Cipriano. John is the Global Head of Cell and Gene Therapy at SK Pharmteco and has over 20 years' experience in cancer cell biology and over 15 years in clinical research in cell therapy. Cedric joined the company in 2015 and currently holds the position of Innovation, Analytical and Process Development Director. Prior to joining the company, he spent 15 years working in the medical device field. Dana, the Global Head of Testing and Analytical Services, has a 20-year career in biotechnology and cell and gene therapy, including leadership roles in laboratory operations and project management. If you would like to ask the speakers a question about this Fast Facts, simply use the Ask a Question box to the right of the viewing pane. I'll now hand it over to John to begin the presentation.
Hi, my name is John Lee. I am the Global Head of Cell and Gene Therapy here at SK Pharmteco, and today we'd like to talk to you a little bit about our platform processes and how they may help you facilitate your clinical development of both viral vector as well as plasmid manufacturing. The title of our talk today is to accelerate time to the clinic for advanced therapies with a platform approach. Before we get into the science that underlies our platform, let's first talk a little bit about SK Pharmteco as a company. Our parent company is SK, and as you can see on the slide, SK is a diversified conglomerate with over $200 billion in assets and $120 billion in annual revenue. SK is a leader in biotech, chemicals, energy, semiconductors, telecom, and so forth, and is the number 2 conglomerate in all of South Korea behind Samsung. SK Pharmteco, being a subsidiary of SK, has 70 years of external manufacturing experience across 8 different manufacturing sites across the world and has demonstrated 60+% of growth over the past 5 years. SK Pharmteco today is divided into 3 different business units. One of those is the small molecule business unit, which as you can see is across South Korea, the United States, and Ireland, and then the cell and gene business unit, which is in both United States and in Paris. In my role at SK Pharmteco, there are 3 things that every customer asks about. They want to know, how can you save me time? How can you save me money? And more importantly, how can you continue to provide me with a level of reliability that I would expect from an external partner? We'll get to the reliability piece in just a moment, but first let's talk a little bit about time and money. Platform approaches really enable the savings of both time and money. In terms of money, we're saving up to 40% through improved supply chain channels, the elimination of unnecessary or even open unit operations, increased internal know-how associated with that platform, and streamlined logistics associated with the platform as well. So when you bring all of these things together, we're able to then cut out the number 1 cost driver in the GMP facility, and that's the human element, and then also really leverage those supply chain channels in such a way that we're able to procure savings and then pass those savings along to our customers. On the speed aspect, we're looking at shaving about 6 months, a little over 6 months actually, from a more traditional CDMO partner relationship due to those integrated activities that we're able to do under one roof, including things like process development, the tech transfer, and then all the associated downstream analytical work, being able to really do those concurrently in such a way that we can then really streamline the clinical development process. A big component of enabling a platform is the cell line. The availability of a robust cell line that's capable of producing complete, highly infectious particles is one of the number 1 things that will enable that platform. At SK Pharmteco, our platform processes are underlaid by a fully owned, fully proprietary cell line that enables both lentivirus as well as a variety of different serotypes of AAV virus manufacturing. The lines are fully GMP banked and importantly come with zero licensing fees for any customer manufacturing with us. Furthermore, we've done the analyses and we believe that we can save most customers over $1 million per batch in pass-through costs when they hit large-scale commercial GMP manufacturing. And so the cell lines that we have, we're very excited about. You won't see any data today on those cell lines, but we will share some data with you in terms of the output. And that's going to come to you next in the next section through Cedric Rousseau, our head of process development at SK Pharmteco Europe.
Thank you, John. Now I will present you our viral vector manufacturing platforms. We have two platforms in place in GMP manufacturing suites. The first platform is dedicated for lentivirus manufacturing. Its name is Lentisure. We are able to produce vectors with adherent or suspension cells. The second platform, called AV Velocity, is used for AV manufacturing with cells in suspension. Those two platforms are plug-and-play, proven and robust. I will illustrate those arguments in the next slides. For a product point of view, we are able to release a clinical batch 12 months after receiving the plasmids. Our two platforms are robust and highly scalable. The graph here shows that the titers obtained with Lentisure or AV Velocity are consistent whatever the batch size. The productivity at cell culture harvest in a 250 ml shake flask or in a 200 liter scale bioreactor are the same for a specific vector. We can achieve high titers with both manufacturing platforms, up to 1.9 e to the 7th infection genome per ml for Lentisure, and up to 2 e to the 11th viral genome per ml for AV Velocity. Let's focus now on AV Velocity platform and its performances. We can obtain up to 80% of full particles. The manufacturing process is highly productive. The total yield is between 30 and 50%, that is quite high, and it's possible to obtain a final titer at 1 e to the 14th viral genome per ml. Moreover, the scalability is proven up to 1000 liters. Now let's talk about our track records. We have a huge experience with those platforms. 100 batches of Lentisure have been made, including 40 cGMP clinical batches, with a total of 14 different transgenes. With AV Velocity, more than 100 batches manufactured, 55 cGMP batches released with 5 different transgenes. Now I'll let Dana Cipriano, our head of Analytical Business Unit, to speak about the analytical platforms.
Thank you, Cedric, for the introduction. My name is Dana Cipriano, and I am going to speak with you today about the SK Pharmteco analytical platform strategy. One of SK Pharmteco's key differentiators for our clients is our analytical platform strategy. The platform is focused around 3 pillars, with the first being capacity. SK has global analytical development and GMP testing laboratories to support the capacity needed to meet our clients' demands required for cell and gene therapy products. The labs have been purpose-built using a modular design that allows SK Pharmteco to scale capacity ahead of testing constraints. And since our labs have been purpose-built, this has allowed us to co-locate testing operations with manufacturing, as well as having testing on the manufacturing floor for in-process critical assays where possible. SK Pharmteco also has a full menu of testing services, allowing for an end-to-end solution with one company. This includes in-process, characterization, and lot release testing, in addition to raw material testing for manufacturing. Many cell and gene therapy clients are required to submit testing to many different laboratories, creating project management and scheduling challenges. Having all services within the same campus or across the SK Pharmteco network allows for one point of contact and a reduction in the amount of product required to test batches. This coordination and flexibility allows SK Pharmteco to offer accelerator programs that can reduce and eliminate weeks from the analytical testing process. In addition, we recommend to our clients to start characterization of advanced therapies as early in the drug development life cycle as possible. Using our advanced analytical capabilities, such as sequencing, mass spec, and potency assays that are specific to a client's product, allows for early testing that can support comparability testing later in development or if changes occur for improvement of the manufacturing process. We also offer stability programs to evaluate product and reference standard stability throughout the product lifecycle, as well as a full CMC regulatory team to support our clients during the regulatory filing process, enabling approval of medicines to patients faster. Finally, innovation. SK Pharmteco understands the importance of being able to meet the complex analytical challenges, so being able to continue to invest in new technologies is critical. Having orthogonal methods for early product characterization is a main component of the testing strategy. SK Pharmteco does this through continuing to expand into new technologies, having collaborations with world-class partners, and continuously investing with our sponsored research agreements and the experts in the industry. The accurate characterization and testing of cell and gene therapies is mission critical for the industry to minimize impact of regulatory delays. To bridge this gap, developers need access to services and tools that allow for sensitive and specific assays with the flexibility to test across the 5 key domains required by regulatory bodies: purity, potency, identity, safety, and stability throughout the manufacturing process and product lifecycle. Our platform assays are defined as an analytical procedure that is suitable to test the quality attributes of different products without significant changes to its operational conditions, system suitability, and reporting structure. This type of analytical procedure can be used to analyze molecules that are sufficiently alike with respect to the attributes that the platform analytical procedure is intended to measure, and are typically qualified or validated at SK Pharmteco. This helps to accelerate these complex methods for product-specific purposes. At SK Pharmteco, we have a comprehensive AAV method platform package available not only to support our CDMO clients, but also the testing programs to support external or standalone testing to reliably characterize and release AAV products. Sample volumes for testing remain a challenge for manufacturers, as a significant portion of the GMP batch goes to analytical testing, making less product available for patients. By having all of the analytical assays available within an SK Pharmteco, this allows for coordination of testing and the most effective use of the product. Some of the longest lead assays required are the safety assays, in particular the replication competent virus assays, as well as the adventitious agents testing. These methods are required by the agencies and are also the longest lead method in the release panel. By having these methods in-house for our customers, it allows for capacity management and reduced release times for our clients. SK Pharmteco has these methods available now and ready to support all AAV programs. Similar for lentivirus, SK Pharmteco has many of the current assays required as a platform service. As with the AAV, assays for lenti are just as complex, requiring orthogonal methods and technologies. While the equipment and know-how of running these assays may be somewhat similar or comparable to AAV, many assays, such as cell-based assays, have different endpoints and readouts, such as the use of flow cytometry for infectious titer assays, which can be supported at SK. As SK Pharmteco has continued to expand technologies for lenti, we will also be able to offer safety testing for cell-based, replication-competent lentivirus testing GMP in the second half of 2024. Finally, SK Pharmteco's approach to assay development and testing is centered around the design and execution of complex platform assays that can support the many modalities of cell and gene therapy. This includes gene therapy, gene-mediated cell therapies, and nucleic acids such as plasmids. Other modalities are also being supported as our capabilities expand. Our scientists first identifies the assays that are required to meet global regulations. Experiments are designed and executed in our state-of-the-art analytical development laboratories with some of the most cutting-edge technology and innovations used to characterize advanced therapies. SK Pharmteco then executes phase-appropriate assay qualification or validation utilizing representative sample matrices that are produced at our facility by our vector engineering or process development teams. Assay qualification or validation is then executed by the technical subject matter experts in partnership with the GMP testing operations that will perform QC release testing. A formal transfer of the method occurs to the GMP testing operations team that involves the training of our analysts, hands-on in the laboratory for GMP characterization and release testing. These platform GMP assays are then ready for testing for our client programs.
So now that Dana has spoken to you about our analytical services, I really want to wrap up today's discussion by telling you a little bit more about what we're doing in the plasmid space and why we're so excited about it. We proudly offer both products and services under our plasmid umbrella. For products, we have over 20 different AAV plasmids, as well as all the requisite plasmids you would need for lentiviral manufacturing. And not only do you have access to those when manufacturing with this, but you can also order these plasmids through our e-commerce platform for use in your R&D labs today to really set you up for success in the long term. So you can use those in the R&D setting today, and as you develop your data packages, eventually you can come to us when you're ready for clinical development and have that really streamlined, seamless transition from R&D into the clinic. As far as services, which are outlined on the right-hand side of this slide, our team offers strain selection across multiple different E. coli strains, as well as downstream cell banking to enable quick amplification of your desired plasmid when you're ready. And then above and beyond the process that you're seeing on this slide, we also have over 20 downstream analytical methods ranging across a variety of standard offerings from stability to next generation sequencing to really get to the quality that you're expecting from a partner providing this type of service. So in closing, I just quickly want to touch on one aspect of SK Pharmtechco's service offering that wasn't covered, and that is the GMP cell therapy manufacturing offering. And when you think about cell therapy, particularly in the world of CAR Ts, you're starting with a plasmid drug substance starting material, which is then feeding into viral vector, and then those viral vectors are then coming over for gene-modified cell therapies. And so when you come to SK Pharmtechco, you literally are getting that end-to-end service offering that's depicted on this slide, starting from plasmid, going on to viral vectors, hitting that then cell therapy drug product on the back end. Each of those GMP services is supported by an extensive in-house analytics team, as well as a dedicated quality team that have years and years of regulatory experience to ensure that there are no hiccups on the regulatory side. And so as you think about SK Pharmtechco, we ask that you really think of us as a true end-to-end service provider that is offering all the things that you're going to need to be successful in the clinical space, in cell and gene therapy, and we look forward to hearing from you. With all of that in mind, I would just like to thank you for your time today and would direct you to our email address that's depicted on this slide for additional questions and or to be contacted from somebody on our business development team. And that address on the right-hand side of the slide is skpharmtechco@sk.com.
All that's left is to thank our speakers, John, Cedric and Dana, for a fascinating presentation. And thanks to you, our audience, for tuning in today. If you'd like to download a poster version of today's talk summarising the main points, you can find that below the video. We'll hope you'll join us again soon for more Fast Facts.